Hi, Jordan. Can you see me? Can you hear me? Okay, now you can hear me, right? Good, that makes things much easier. Okay, what's your question? Oh, let me see. I want to unmute you. Participants. Okay, I should be able to hear you now. I mean, yeah, can you speak? Oh, okay. Yeah, go ahead. Type your question. It's so strange to talk with no response. Okay, chapter 10, number 4. I have to pull up I should have printed it. I'm going to print mine so I can look at it without changing screens. Okay, let me see. Number 4. Oh, yeah. So this is the Mechelson-Stahl experiment. And let me see. Hold on. I'm going to change my share screen. Okay, you should be able to see my notebook now. Can you see it? Okay, cool. All right, so here's the thing: it's really a pretty neat experiment. So bacteria can grow on very limited nutrients. So if you get the right kind of bacteria, they can live if their only nitrogen source is ammonia. So you give it ammonium sulfate or ammonium chloride, and that's the only source of nitrogen they get. And so if you feed bacteria N14 ammonium chloride, all the nitrogen is going to weigh 14 grams per mole throughout the entire organism. You could alternatively feed the bacteria N15, and that's going to have a different mass. So this will be heavier. This will be lighter, the N14. So they grew the bacteria, they isolated the DNA, and the nitrogen bases in the DNA have the nitrogen. And so let's say we have and they're going to have different densities. So they grew the bacteria, they isolated it, then they centrifuged it. Remember in the chapter when we talked about centrifuging as a way to separate organelles in cell lysis? Do you remember that? Okay, cool. Okay, so imagine all of the DNA. They grew the bacteria for several generations. And this is actually kind of the whole like logarithmic growth and generation time is sort of relevant to the viral replication we're looking at now in patient transfer. But so if they grew the bacteria for several generations, they've got parents, grandparents, great-grandparents, all on a nitrogen 15 medium, the density of the DNA would come out in the sedimentation when they centrifuge here. Okay, now imagine they take that bacteria, they spin it down, they throw away all the media it was growing on that had N15 in it. Now it's going to have N14, and the bacteria grow for several generations with the lighter ammonium chloride as its only nitrogen source. So they grow it. Now there's 3-4 generations only with the light nitrogen. When they do the same experiment, the DNA is going to be less dense. And it will not sediment as far down in the tube as the N15 grown DNA. You with me so far? Sorry, I'm losing my... Okay. So now here's the tricky part, and it's sort of amazing that they did this. But so now imagine you start with either the light or the heavy nitrogen source. And for one generation, you allow it to replicate once, maybe twice, and you isolate the DNA and you do the centrifugation. What will happen is that you get a band that's halfway in between. So what this represents is down here, you have 2 strands of DNA. You have 2 strands of DNA that are N15, and it has that density. Up here, you have 2 strands of DNA that are N14, and it has that density. Intermediate, you have 1 strand that's N15, and you have 1 strand that's N14, and so its density is halfway in between the N14 and the N15, which means that it had 1 old strand from the previous media and 1 new strand from the new media. This would not be possible unless one of these was a parent strand and one of these was a daughter strand. That's the only way you could have something that's in the middle and only in the middle. Does that make sense? Okay. What else can I clarify about this? Basically that when you do DNA, when DNA is replicated, you have part of the old and part of the new together. That's the term semi-conservative. So you have 1 parent strand, 1 daughter strand. That explains how the copying is done, because it knows that it has, the parent strand is the template for the daughter strand, and the parent strand, the template, directs the sequence of the daughter strand. Cool. What next? Okay, no problem. Oh, number 10, let me see. Oh yeah. They all have exonuclease function. Hold on. So Do you know what exonuclease activity is? So exonuclease, this is over there. We go a little higher. So exo, the outside, nuclease means it's hydrolyzing nucleotides or breaking apart nucleotides. Let me see. Can you see that? Okay. So imagine, I'm going to draw this. You have a 5' to 3' strand of single-stranded DNA. Five prime exonuclease activity would be removing this base. It removes the bases one at a time from the 5' end. Um, 3' exonuclease activity is removing nucleotides from the 3' end. Okay, so both of these activities are required in DNA polymerases. All three of them, there's DNA pol 1, DNA polymerase 2, this is in bacteria, and DNA polymerase 3. They all have 3' exonuclease activities, which is kind of weird. So imagine, let's see, my template's up here. And so synthesis is going in this direction. And so what happens is the polymerase goes in this direction and then comes back, goes in this direction and comes back. It synthesizes, all of them always synthesize 5' to 3' activity. And they all have, I may have misspoken, they all have 3' exonuclease activity. So they go this way, they go this way, and then they might take a few out with the 3' exonuclease activity. The 3' exonuclease activity is associated with proofreading to make sure that the bases that are put in are correct. It's also associated with DNA repair. So if there is a mismatch with the template, there'll be a distortion in the DNA structure, and then the repair mechanisms go into place. And in order to repair a mismatched inserted base, they have to have exonuclease activity. So they all have 3' exonuclease activity. DNA polymerase 1 has 5' exonuclease activity. It's the only one that has 5' exonuclease activity, and this is associated with removing the primers and replacing it with nucleotides. Does that make sense? Do you have more do you have, is there something, oh, let me see. Okay, so you're fine on number 10? Okay, good. Sorry, I don't know when to stop because it's so quiet on the other side. I found quite a bit of differences between 1 and 3. Let me look at the question. Oh, let me go grab, I printed, let me go grab it, I'll be back. Oh, okay. So yes, there's a lot of differences. I would say the most important things are it's not so much the size of the enzyme, it's what their activities are, their efficiency, and how that relates to their function. So which polymerase is the one that does the majority of the replication of DNA in bacteria? There's DNA pol 1, 2, and 3, right? Which one's responsible for copying the genome? Yeah, and so why? Like, what makes that? What properties or what capabilities does it have that suggest that? Okay, it's fast. It synthesizes really fast. Which of the elements of that table tell you that it synthesizes fast? No, close, very close. And that processivity is not about speed. It is an important factor as to why DNA polymerase 3 is thought to be that copies the genome. Processivity is literally the polymerase is on the template and it's making the synthesis. It's how many bases does it make before it comes off the DNA, off the template. So for DNA polymerase 3, the processivity is 500,000 nucleotides. That means it puts down 500, 500,000 nucleotides before it falls off the template. That doesn't say anything about speed, though. It does correlate because there's an efficiency there, but the speed is kcat, the turnover number. That tells you how fast. But those are the two factors that you look at to tell you that DNA polymerase 3 is the polymerase that copies the genome. Those are the two. So it's turnover number and processivity. Okay, now DNA polymerase 1 is associated a little bit with repair, but the unique function that it has is the Nick translation reaction. Nick translation, that is what happens when you remove primers and replace it with DNA. So imagine you've got, so this is DNA polymerase 1. You've got some DNA. Let's see, this is 5 prime, that's 3 prime, 5 prime, 3 prime. You know that DNA polymerase needs a primer and a 3' hydroxyl in order to synthesize. So here's my RNA primer, and then DNA, and that was put down by primase, put down the primer, and then DNA polymerase 3 came in and made DNA and attached DNA. This is RNA. This is my primer. Primer. Okay. So this is a problem because we don't want RNA in the genome. It's not as stable as DNA. So what happens is DNA polymerase 1 comes in and it removes the primer of RNA. And the primer can be either RNA or DNA, but it removes the primer, and it does so with 5' exonuclease activity. This is the only polymerase that has 5' exonuclease activity. But then it uses its synthesis activity to replace the RNA with DNA. Primers are relatively short, so DNA polymerase 1 doesn't need to be highly processive, and it doesn't need to be very fast, but it needs to have the 5' exonuclease activity, and it needs to have the DNA polymerase, the DNA synthesis activity. So those are the important features that you look at in that table to identify, and this is literally how they did it. That's how they figured out what function those polymerases had in the cell. Does that help? Where is synthesis activity of DNA polymerase 1 shown? I don't know what that means. On the table? Oh, well, they all have, let me see. They all have DNA synthesis activity. Let me see if I can find the table in the book. Oh, when it says polymerization 5' to 3'. And the fact that it's a DNA polymerase. These are good questions. More questions? Oh yeah, no problem. Also, if you, I can hunt for more videos about, like I'm thinking a video about Nick translation reaction might be a good idea. Let me know if you think that there's a specific topic that I should try to find some more depth or something to look at that would help. Oh, I think writing a lot is good. I actually think the more you write, the more time you spend in the material, the more you're going to absorb it and soak it in. Number 20. Ah. Okay, so for number 20, I actually think this is a good one because you want to, what I would say, I was just actually sitting going over the questions myself. And what I think is a good thing to do is make a list of what you need and then try to remember what it's for, right? So the things that are required for DNA synthesis, here's how my head works. Well, of course, we need a polymerase, and that synthesizes the DNA. DNA polymerases all require a 3' hydroxyl, and so that is provided by the primer. So I would, and I think a good way to look at this question is to make the list and then make sure that you know what each part is for. Okay, and then of course we need the nucleotides. So we need DATP, DGTP, DCTP, and DTTP. We should have magnesium. I'm not as worried about that. We need, let's see. Well, if we're talking about the replication fork, we need DNA gyrase. That's a type 2 topoisomerase. We need that ahead of the replication fork to relieve supercoiling strain. Once strand separation takes place, we need single-stranded binding protein to keep the DNA strands apart in the replication fork. And it also protects the DNA from being hydrolyzed, from being broken apart. Okay, you're good on that? Okay. Yeah, I would just go through the list and then make sure you understand the function. Sorry. What else? Once you get to the eukaryotic stuff, it's pretty overwhelming, huh? Oh, good. Okay. Well, I just, yesterday I uploaded a video for transcription and it was so, I was torn about whether to bring it up because it was a really good video, but they have the incorrect title. They said it was transcription in prokaryotes, but it was transcription in eukaryotes. So I put that in the PowerPoint so that you guys would know that. And then I actually thought, well, here's a good question. How do we know it's eukaryotic transcription and not bacterial? But that's next chapter. SOS response? No, don't worry about that. Did we didn't have a question on that, did we? Number 41, let me see. Oh, no, don't worry about the SOS response. Yeah, that's what it's related to. I mean, I think you should know that without DNA repair mechanisms, organisms are not viable. And I mean, it's actually pretty stunning. If you are missing one of the DNA repair mechanisms because of a gene mutation, people don't live past infancy or toddlerhood. That's how much we rely on the repair mechanisms to um, maintain the fidelity in our genetic material. How can the SOS response or other ones? There's multiple repair mechanisms in both prokaryote and eukaryotic organisms. Does it say better? I don't think of one as better than the other. Um, we have base excision repair, nucleotide excision repair. Um, what are the other ones? There's all these different mechanisms. There's direct repair, like for thymidine dimers. Base mismatch repair. Does it say better somewhere? I mean, I think it's different because in a prokaryote, you're just dealing with one cell in an organism. Yeah, okay. Oh, yeah, I see. Okay. $51. Oh, okay. So for 51, what I would say is it's in terms of the differences, know the major themes. You don't have to know the details, but for example, replication begins in one place at OREC in prokaryotes, but in eukaryotes, there are multiple replication forks going at the same time. Um, on top of that, in eukaryotes, the DNA is packaged in nucleosomes, right? So the DNA in eukaryotes, right? So they are in nucleosomes, and the DNA is wrapped around histones. So before DNA replication can be initiated, the DNA has to be made accessible. And so there has to be unpacking of the nucleosome. So there's multiple replication forks. And there's also DNA unpacking. And the other major thing is that DNA polymerases in eukaryotes do not have exonuclease activity. So I would say the big one is the multiple replication forks, the histones, and the lack of exonuclease activity. But yeah, the processes are very different. The names of you want to know generally the different kinds of polymerases. Number 56. Yeah, it would stop. Yeah, the telomeres are pretty interesting. There's some correlation between the length of our telomeres and our longevity. Cool. Well, it sounds like you're on top of it. Suggestions for an additional video that I could find that would be helpful? Yeah, there is, but not all of it's good. So I like to be able to vet them a little bit. Anything on Khan Academy is reliable, but they tend to be kind of long and tough to sit through. There's some other ones that are a little bit more sophisticated, but sometimes they have mistakes. That's a good idea for the Khan Academy. That's a really good. Can you still understand what he says? I don't. I have to look and see if I can figure that out how to do that. Good. Well, I think this is going to help you for your MCAT. Oh, my pleasure. Okay, so then I guess plan for Thursday to talk about transcription, and we hopefully we'll get a cohort then. Take care. Oop. Hmm. Case review. I c- Probably I'm seeing it's going to be hard. I'll see what I can find. I feel like the workload is about appropriate with this. What do you think? If I can find something short that focuses material with the three different class preps moving on to online, I'm treading water, keeping up. Yeah, well, yeah, I actually think I miss having people in class, to go back and forth. But I feel like this is actually an okay format for this class because you really just have to spend a lot of time with the material. Okay. Okay, well, I'll see you Thursday.